All right. Minasan konnichiwa. Hello, everyone. My name is Lonni Gomes, and today it's my pleasure to share with you some progress that I've made in some research trying to look at the quality structure of color. So,
Lonni, maybe you can minimize the Zoom style.
Sorry, I cannot hear you really well. There's a bit of an echo.
You probably are opening the participant's view to the right, right? Yeah, can you minimize or remove that?
Sure. Is that better?
Yeah, that's better.
Sure. I can't see you now. So you'll have to yell out if there's any question. So the starting point of this research was, can we try to see how the color experiences that we experience every day, how are they organized in our mind psychologically? And there's most likely a structure, and can we look into that? So we looked at two models in this research. The first one is extremely popular. It's called the RGB model. And another one is a much more sophisticated CIE lab. And both of these models, they are both using three-dimensional representation to organize the color. And so our research question was, is the color experience, the qualia of color, actually accurately captured by these existing color models? And to do that, we used 93 colors, and we asked participants to rate the similarity of pairs of colors, and each participant rated 324 pairs, including a double pass paradigm to try to test the reliability of the similarity ratings. So using all these similarity ratings, we generated a similarity matrix. And so how to rate this figure is by looking at a color on the x-axis and a color on the y-axis. The intersection of these two provides the similarity rating. So what we can see here is the main diagonal dark. It's when the color are the most similar, and that's usually because the same color intersects, and color that are indexed to the side are also quite similar. We can also notice that there is in the corner around here, and I don't know if you can see my mouse, but around here we can also see a higher similarity concentration. And exactly, thank you, Noel. So now that we've seen this similarity matrix, on the next slide, I'll have the same repeated for also the RGB model that I mentioned earlier, that we can see on the third slide, on the first line, and the CIE lab model on the right-hand side. I've also included data from one of my colleagues that looked at similarity ratings among colorblind people, mainly red-green color deficiency. Yes?
Can you move the Zoom panel to the bottom, or not that one, the one on the left? Yeah. Can you move that to somewhere like below? It's blocking the title.
Is that better?
Yeah, that's better.
Okay.
Yes.
Okay, so and on the second line, starting from the left panel, it's the data that I mentioned earlier, but this time it has been shuffled to create more of a random data set. And also on the second line, we have a panel which is still the RGB model, but where I added some Gaussian noise. And similarly for the lab data set, we also have the same similarity matrix with the inclusion of some noise. So now I've presented the different data sets that are going to be used to be compared. So first, I've generated some multidimensional scaling to see how the data was organized in a fairly conventional way. So for the normal color vision people, we can see a ring-like structure emerged, while for the RGB and the CAE lab, we see a much more diffuse and homogeneous representation of the colors. Unfortunately, I did not get the MDS, the two-dimensional MDS for color vision deficiency. I apologize for that. And for the second line, we can see that we lost all structure with the shuffled data. And when we look at the RGB data set and the lab data sets, the inclusion of noise did not disrupt the structure either. Now, I would like to introduce a few concepts from the topological data analysis approach. One of the first critical aspects is that the exact shape of an object is not necessarily important. What matters is the presence or absence of a hole. So here we have 4 objects that are all considered equivalent for topological data analysis because they have a single hole in the middle. And if we now look at different type of object, we can see that if we start from this circle here, now if we have a full disk, these two are not equivalent. But if we look at a thicker with still a hole, that's equivalent. And this almost figure 8, there's also, once again, only one hole. So that's an equivalent object. However, if we have like a full figure 8, we have 2 holes, and that makes it a different object. This idea of holes is really how in topological data analysis, we're going to try to categorize and separate objects, which will have importance in the next few slides. So the main takeaway here is when we look at objects in topological data analysis, what matters is the number of holes. So now how do we apply that to data? So on the left-hand side here, we have all the different points. And here, in our case, is going to be the 93 colors. We all have them organized, and that's going to be based on the similarity ratings. And what we're going to do is we're going to run an algorithm that is going to slowly increase the diameter of each data point and start to notice when points are starting to touch and connect to one another. And this connection is what is going to start creating the objects that we had on the previous slides. And we're going to be interested to see when holes appears in these objects and also when they disappear in these objects if they start filling up. So if we look, for example, here on this one, we can see that the points here are all touching, but there's still the presence of a hole between these 4 points here. And so that's why we have this object here. However, when we look at further down, they started to completely fill this space. And so there is no longer a hole. And I've also included here some extended presentation of how data points can start to connect and how we're gonna be interested as we increase the diameter of each data point, how the nature of the object and the presence of different types of holes is gonna be of importance for us. Okay, so now we have what is called barcodes for topological data analysis. And on the x-axis, it's sort of a timeline. This is the size of the increase of the diameter. So as diameter increases, we can see that some holes are appearing immediately, but we can also see when the holes are disappearing. The y-axis is arbitrary. It's just to stack all the objects that were mathematically identified during topological data analysis. So for the RGB data set, we have a very conventional result. We can see that we found three types of holes, and we're going to consider this that the data set is three-dimensional in nature. Now, if we look at the CIE lab, it's a bit hard to see because it actually is a single dot here, but we actually got an extra dimensional object in the CIE data set, which in later slides, we may have some explanation for. Now, it becomes more interesting is when we look at the experimental data for normal color vision. We found quite a fair amount of four-dimensional, but even at five-dimensional holes. And an important aspect is that there may be further dimension. I stopped at 5 dimension purely because of computing power. I was limited by my machine, and hopefully I will be able to push this analysis further using more to look where really this data set can stop. And similarly, in color vision deficiency participants, we can see that there is still a high number of dimensions, even though we can also notice that the amount and structure of the holes has significantly changed compared to the normal color vision. And if we look at the two RGB and lab data sets with nose included, we can see that there has been very few four-dimensional holes that appeared in the RGB dataset, and a few extra holes that appeared in the lab with noise dataset. However, there are still differences compared to the normal color vision. And when we look at the shuffle data, there are a high number of dimensional holes. However, the structure compared to the normal color vision is totally lost. Is that serious? Sorry, I lost you for a second. Can you still hear me?
Yeah.
The screen suddenly cut. So we can see that using random data, we still have a high number of dimension possible. So that's why we're also interested in looking at the structure of the holes and when they appear. Now, on the next slide, I have represented the same data, but in a format that is sometimes more used for topological data analysis. So it's exactly the same results, but this time this is persistence plot, where instead of having bars that show the start and the end of the holes, we have on the x-axis when the hole appeared, and on the y-axis at what time it disappeared. So instead of a bar, we now have just a single mark, which allows us, for example, for the CIE lab to see the single dot in fourth dimension here. Okay. Now on this slide, I have included some testing that I've done on each data sets to look at the frequency of triangle of inequality violation. When we look and we talk about distances, as we have in the past with spaces, it is important to verify if the triangle of inequality is violated or not. And unsurprisingly, if we start with the RGB, there is zero triangle of inequality, which is normal considering that the model was built with a 3D dimension and organizing the colors in a way that is, say, Euclidean. However, for the CIE lab, we have a high level of triangle of inequality violation, especially around the green and blue. And this, we believe, could be the reason why we found a single four-dimensional hole in topological data analysis. However, if we look now at the normal color vision on the top left panel, we see that there is some triangle of inequality, but where it's located is very different from the CIE lab and the color vision deficiency people. And we also see that there is a high level of triangle of inequality violation in the shuffle data sets, which is to be expected, considering that the shuffling would not attempt in any way to preserve this axiom. And if we look at the RGB and noise, we can see that adding some noise to the RGB data set starts to elicit some triangle of inequality. However, the distribution appears fairly random, unlike the normal color vision, where we can see these two parallel diagonal forming. And this is also of interest because in other research, I've also found a violation of asymmetry, which I could get on if there is interest in the audience. So sorry for the flashing of the slides. But that's all I had for today. So I'm now happy to take questions if I'm sure there will be.
Maybe Lonni, you can probably present the next slide.
Yeah, next slide.
Yeah.
Okay. So this is quite a busy plot, but if I can try to walk you through it, what we have on the x-axis is the color that was first presented to the participants. Earlier, when I presented the methodology, colors were presented one by one to participants for each pair. So they first saw a color, then saw another one. So on the x-axis, we have which color was presented first, while on the y-axis, we have which color was presented second. Now the color within the matrix, this is the level of asymmetry. That is, if the participants rated the color pair as more similar or less similar, depending on which color was presented first. That's another axiom in distances, is that going from A to B is supposed to be equal from going from B to A. However, this plot indicates that there is a level of asymmetry that is also appearing along this diagonal around here. Thank you. Now, which is consistent with what we've seen in the violation of triangle of inequality.
Okay. Do you have any conclusion slide or?
Yes, conclusion. What we have is that unlike the conventional model would have us so far, it seems that looking at color requires us to go beyond tridimensional representation. And if we're looking to qualify the Qualia structure of color, we have to look at multidimensional. And we have seen 5, but there is also research that have shown that like probably 7 could be also a good, using totally different techniques.
Okay, thank you. Question, comments?
Okay. Thank you for the very interesting talk. I'm curious about the violation of the triangle inequality. Could you show the slide? Yes. First, can you explain again the meaning of the matrix?
Okay.
Triangle equality itself contains at least 3 points X, Y, Z.
Mm-hmm.
How can I interpret that?
Yes. So on this matrix, on this all matrices, what we have is the proportion of violation. So if we take 1 point and we look at the x-axis and y-axis, this forms a pair. And for this pair, I've calculated how frequently the triangle of inequality was violated looking at the 91 other colors.
Okay. So the points shows the pair X, Y. And comparing this and the third point Z, and the frequency of the violation is graph of Y.
I'm sorry, the sound was getting a bit echoey towards the end.
Now, it's Y and yeah, is bigger than this, so that's a probability. The probability of this for all Z, over this 91 hours. Okay, so XY is denoted as the position of the matrix components, and the black means the probability of violation.
Right.
Okay.
So what was any color? If this is blue, then that's very black.
Okay. So the next question, can you hear me?
Yeah, better.
The next question is why black noise can violate? Because my understanding is correct, RGB itself is within the Euclidean space, and IE may be different. No, it's not. CIE is not embedded in the Euclidean space. No. Okay, so then I understand, but what kind of manifolds?
Ronnie, maybe you can show the first slide.
The first slide.
Introduction to the RGB.
This one?
Like embedded in three-dimensional space, but it is not.
So the CI is has a correction to make the progression from color to color more homogeneous, and that's around the green blue.
So it is not a metric space. CI lab is not a metric space.
Kind of correct.
Going to the metric space more.
Right. So you almost like an RGB specification to pick a point.
Yeah.
But to define the similarity in the two, then depending on where it is, it has some kind of weird bending, right? And that's supposed to kind of equate the similarity or similar to human, according to this formulation. But as you saw, it's very far from human actual similarity.
Actually, not actual similarity. So it's just a modification of RGB into the more human-like, but actually it's not from your study.
From my
Your study. And so CI is some kind of the not Euclidean, not Riemann manifold, Riemannian manifold. It's Lorentzian or something.
Yeah, if I maybe I am wrong about that. I think it is supposed to be Riemannian.
But
there was one paper in PNAS that appeared like last year or so, saying that color manifold is actually not Riemannian, unlike we did for 120 years ago.
not Riemann.
Slightly friend us about the manifold of CI lab, not the human possible.
CI lab is probably
But
Riemannian.
the Riemannian has some metric.
Yeah, the to A to B is like a minimum. Or something like that.
But then why it can be violence?
I don't know. I may be wrong.
Okay, but
Do you know?
The CA lab,
Oh.
the difference. Our student CA lab concept. And as in CIE lab stage, it's three-dimensional, but the line-axis is red and green. The another axis is yellow and blue. Or axis corresponds to the brightness.
That's a 3D. So actually, yeah, that's one thing I forgot to explain. But that kind of a three-axis, primary axis, it reflects the human
Yes.
perception. So green, yellow, and then luminance is another configuration.
It's based on color of the theory.
But the matrix, there is no well-defined matrix on that.
I think by default, the 2 points, the distance is almost like a distance.
Almost. But not.
When it's really far, for some reason, they do some kind of correction.
So it is known that in CIE, the violation of triangle inequality, right?
I actually don't know. You know?
I don't think
I think it would be known because they use different way to calculate distance depending on where you are.
Okay. So the point is, in that sense, CIE seems to be more similar to actual phenomenal similarity, but your study makes it clear it's not so very interesting. And of course, maybe you know about quantum cognition. In quantum cognition, this kind of the violation of triangle inequality is very good. Very hard to say hot stop one of them. So maybe it's also suggests maybe some kind of quantum cognition like model is more accurate. Yeah, it's just a comment.
Do you think that once again, Lonni showed that Lonni, can you go to again to triangle inequality violation first? Yeah, so here you see that this part of figure around here, and around here, top right, that's a real data. Yeah, yes. This combination tends to produce a lot of triangle violation. With respect to another third color. And then Lonni, go to the next one. The asymmetry. And here we see asymmetry tend to happen around here. Is there a theoretical thing that we haven't seen the asymmetry or the similarity and triangle inequality to cover the same here? I don't know. If there is just an empirical,
Yeah, because we have the main general metrics space pitch satisfies triangle inequality, but not unbound and also the context. So logically we depend. But yeah, it's very interesting. Why each kind of mechanism? Maybe the quantum cognition like model can solve that kind of situation. Like if the axis is so if the axis is positive, then the projection is commutative. But if the axis is so how to say this kind of known also cases, projection can be conformative. So such axis has maybe some angle, and this kind of orthogonality violation of the basic axis, violation of orthogonality of basic axis maybe and these orthogonal words are similar kind of axis. Cross as of the axis. So my answer is in general, I don't think that the asymmetry and the violation of triangle inequality is logically related. But for example, the quantum cognition model can explain both kind of phenomena and the correlation between us.
you may speak to
the asymmetry happens around blue, the green, the upper red. Is that correct?
yeah, I think so.
I think similar to the hydrogen, I thought analogy. So notice that the red is the primary color. Going back to the previous things, talking about how red is the first. So it is this red, the red related colors well separated. Orthogonal space where as you suppose you have a limited capacity for red colors, and red is your primary color. That's red is your initial, and you devote those. It's similar to what the students yesterday about the V1 is most of the area of the brain, V1 is dedicated over. And then as you go further down the track, it's convoluted. Maybe something analogous is happening here, where most of the neural resources go to primary color, red, allow full orthogonality in space. But you don't have a finite amount of brain in space. As you add in more color, and we call it blue was the last one. By that other speaker we had. It seems rather analogous. As you get red, it takes up like analogous to red, green, yellow, blue. Red, yellow, green, blue is analogous to V1, V2, V3 situation where V1 takes up most of it. You've got plenty of
That's clear. That within V1, by the way. Within V1, this is the formula. And it is a paper.
yeah. Okay, and that's fine. What I'm interested in is asymmetry bias, the structure here.
Primary.
Yeah.
Of low prime.
Yeah. So just that red, yellow, green, blue are the same thing. So you can see that the resources, the red is quite makes available a orthogonal space. But as you move along, you start to run out of resources. Then the orthogonality and given up, and it's been corresponding way, but in asymmetry in the latter. In other words, you put it here, and I guess the foveae maybe the orthogonal region space. Periphery doesn't lose the orthogonality. And imagine you only have a finite amount of brain space. The initial earlier colors you can make it available. But as you start trying to affect it, start to collapse. And once you get a collapse, that's not a orthogonal space. It's kind of resource.
But I don't know how it actually relates to the standard kind of model of the color. Usually, primary axis, the most first one, Z axis is the luminance, like white to black. And then also to it, we get to think about the red axis to red and green axis as an important. And then within that, that's kind of plane of blue, and then these three orthogonal axes there, and then any color is located within these three. That's the sort of default idea in the color cycle, right? Usually. But the first thing is that we, according to this model, if that's true, then any kind of 2 points and the measuring similarity, if that's like a distance. And then correction of the similarity defined in the proportional space, which is more than 5 million. Right? And I'm not sure what kind of axis we're talking about with a limited resource in here.
Well, all this has to be implemented by the brain.
Right.
The brain is the available neurons is the resource. So in the early part of the brain, you've got a huge amount of resources. So you can, for example, the Cartesian product can be easily represented if you've got enough neurons. You've got n by n neurons. The n squared neurons represent all those things, right? But suppose the early part of the brain had those neurons. But as you start to as you start to decrease the total number of neurons, you have to lose some sort of shrinking. So for example, the non-orthogonal one, you say one over k n squared neurons, and that's where.
I don't know whether that addresses your idea, but the most of the color model based on your 50 thing, like in this way, like let's say there is some kind of a neural population that corresponds to degree y position in this position and the degree in this position and degree in this position. Some one upper string result is in combination to locate that what you're saying is located something. So for that, at the minimum, you need all these three neurons, which then cause these things in brain coding. But that's probably implausible, or not really the case for actual neurons. Actual neurons, if you report that, then some of them just respond to the color like only here, or around here, or this region, or if there's any particularly. So let's start thinking about what's happening in the brains before
Maybe related question, but I cannot. It's a little how to say but there is no for me there is no evident reason to assume these axes are orthogonal. It's just a convention, I think. So the assumption. So the distance in this space is nothing to do with the real kind of similar. Okay. So making the connection with this kind of, if you want to do something, we need some non-orthogonal kind of. Yeah. So maybe it is possible to make a quantum combination like.
Yeah. Integral references. Lonni, can you go a few slides? What?
Sorry, which slide?
The MDS slides. Yeah, this is the top left one. Yeah. This is pretty much something that macro means that also repeatedly see, right, with our data. If we do the multidimensional scaling, we tend to see this kind of thing. I mean, this assumption probably is very familiar if it's in the same luminance with the different shades, right? Which is kind of something in this number. So, and many of the psychology experiments are usually done with that way. Because there are too many colors, very different colors. But actually, we use a subset of papers, 93 color sets, and then they do sample all these different lumina. It's very uniform within colors, like that. So I really didn't expect this kind of hole in the middle. The hole or center of this thing should be by default gray, which have a same distance.
So you uniformly sample colors, 93 colors from this 3D space in here.
I think it's like this way and then this way.
Okay.
So if that's the case, then I think gray should be always uniformly kind of related to others in distance. Which is what we see in here and here. The gray is like located in the center.
Yeah. Right.
but that's small. I mean, the part of the issue is that we use the background color as gray. So any kind of gray patch that looks really similar to background is difficult to see. But
In this case, so RGB and LAB are three-dimensional.
yes,
So it is maybe better to plot these three-dimensional actually to compare.
we probably did that. But if we do that, it's part of the program of NVIDIA. It doesn't recover here. It becomes like a kind of slanted. Two-dimensional actually dominates. And it's something to do with the sampling of this color.
Yeah, but you know the model actual truth. So then you can just simply plot the truth, ground truth model.
Yeah.
So yeah, without applying MDS. For RGB, yeah.
Have you done it before? Like plotting all these 93 colors in the innate RGB space or LAB space. Can you hear us?
Paulie, is that a question for me?
Yeah, for you.
Yep. What is it? Sorry.
Have you tried to plot these 93 colors as a dots in the RGB space or LAB space?
I'm sorry, I hear you really poorly. Could you repeat the question?
Have you plotted these 93 dots in a RGB space?
I'm not sure I understand the question now. Like, are you asking if I used RGB to select the colors?
No. Plot these 93 colors within the RGB cube that you showed.
No, I have not. I have not.
Yeah, maybe just simply comparing that the MDS of humans. Is interesting.
Locating these 93 dots in RGB and then
Just compare. Like we observe something like a sphere in 3D space or human MDS. But as you say that original color model, the color sampling is more like not surface of the sphere, rather just uniform sampling of the 3D space. Then the structure is really completely different. Very different.
Actually, in some we also included economic policy. So yes, analysis and the result.
can you share? Sorry. Lonni,
Yes.
stop sharing.
Yeah, sure.
In the analysis using only chromatic colors, and you can see the superior and the superior structure. And when we added achromatic colors in the analysis, but this is what we got. the gray colors were not in the middle of the that's
But this is the distance here is defined as a confusability.
not the naming.
Naming, this. So this means that the zero is never confused with anything else. And so that's why it's very fun.
Yeah. And it's just on data if there's direct similarity judgment of two colors. Maybe the extractive structure can be different. Like
If you can align these, that's in itself interesting.
yeah.
The naming and the expenses. One, this is conjecture, but one source of making this similarity from 3D. But my idea is that it's forward because similarity is both direct sort of perceptual experience itself and also some kind of influence from the categorization.
Yeah.
The categorization is like this way. And if these two are merged, then the categorization can actually contract or inflate locally something. Whereas a very far away thing is not affected. And this neighbor contraction of the expansion probably corresponds to quantum convolution thing. Yeah, probably. Local distortion. And then if you try to capture everything, global and local at the same time, then you need a lot of communication to explain.
For example, my conjecture is if the blue and green line our red and green is very typical and usually considered some orthogonal kind of, then maybe it is very commutative and symmetry or non-violation. And orange and pink something becomes more violent that time. Orange and maybe that's distinctness. Distinct something is more orthogonal like it is not my intuition. If the more and more detailed on the red and very, very close orange.
Some naming data. You see that red is actually very small area, but green and blue is gold.
Big punch. The distinct is there also my
That one. We restrict everything. Is
yes,
the red one
shouldn't violate symmetry and possibly once going back to the previous diagram, we had the two
Lonnie's line.
yeah.
Lonnie, can you share it again? Can you share slide again?
Yeah.
Can you go back a few slides? Similarity matrix. Actually, asymmetry matrix. At the end. Yeah. Go back.
Yeah.
These things around here.
I thought yeah, so I thought it was the effect is the other way. So one foot around the blue, and it had the asymmetry.
Yeah.
The red was very fine.
Yeah.
You showed you outlined the two ellipses.
They seem to be the opposite, where the distinctive colors like red and orange show symmetry, and the lack of distinctive colors, blue and green, show asymmetry.
In fact, what I always wonder about to say that your idea has been kind of pretty dominant actually over the like 40 or 50 years actually. Like prototypical like prototype of the color like red, green, blue, it's kind of category center. Like across like style similarity, so that induces that symmetry. Like red and slightly non-red or yellow and slightly non-yellow and so on. I think that has been proposed by this person who proposed that with Roche. And then several people try to replicate. It's not clear that we can replicate, and we also couldn't. I mean, our experimental found that it's not exactly the same, but it's from this as well. It's pretty clear to me that there is no typical effect. That's very clear, yes. Right. Okay. But it's idea-wise it's nice, but empirically this is much more complicated, I would say.